So today I'll talk to you about parasites and the risks of parasites to the blood supply. Just see if I can move along here. I have several conflicts of interest. So I'm an advisor to Roche on emerging pathogens in malaria. I'm also a member of the Canadian Immunization Research Network, and I'm a co-chair for the ISBT's parasitology subgroup. So today I'll talk to you about the taxonomy of different human parasites. I'll have participants think about why the number of genera and species of transfusion-transmitted parasites is so low. We'll describe the steps taken by blood operators to reduce the risks of transfusion-transmitted parasites to the blood supply. And then we'll explain why some pathogen reduction methods may not be currently appropriate for reducing transfusion-transmitted parasite risks. So throughout the slides, I'll provide some general questions that a donor may encounter as they walk in and undertake the donor questionnaire. Generally, we're screening donors with a variety of approaches, but it's very important to realize that one of the first questions a donor will receive is whether they feel healthy. And a teacher of mine, Jay Keystone from the University of Toronto, used to say that fever in a returning traveler was an indicator for testing that person for malaria. So what is a parasite? It's an organism that lives in a host organism, and it gets its food at the expense of the host. And when we think about parasites, there are three main groups that we think about: protozoa, worms, and ectoparasites, or ticks and insects. And today I'll mostly talk about the protozoa and the ectoparasites that act as their vectors. We have a long history of interaction with parasites. And as humans, we have the possibility to be infected with many parasites in different environments across the globe. As blood operators, we take a variety of approaches to ensure that the blood supply is safe. This includes vigilance and surveillance, donor engagement, which includes questionnaire questions, and you'll see some of those throughout the slide deck. Donor studies, and we'll talk about some of those today. Laboratory testing, including testing for Chagas. Good laboratory processes, and post-donation information, which can lead to look-back and trace-back activities. We also undertake pathogen reduction at this point for platelets, but pathogen reduction can also occur for plasma in the form of solvent detergent plasma, or cone fractionation for fractionated blood products. Recently, as the parasitology group published a paper, we looked at the risk for worms, or in this case, filaria, to the blood supply, and at that point indicated that this appeared to be a low transfusion risk. And so today, we won't talk about those agents. What we'll really talk about are the single-cell eukaryotic organisms, so they have nuclei, that can propagate in humans and cause diseases. So Plasmodium, which causes malaria, or Trypanosoma cruzi, which causes Chagas disease, or Babesia, which causes babesiosis, and ectoparasites, either insects with 6 legs, like mosquitoes, or reduvid bugs, or like ticks, arachnids, they have 8 legs. So those are the two groups of organisms that we're going to think about. The list of agents that are actually a risk to blood supply, when we looked at it in the ISBT group, is quite short. And really the risks are largely going to be confined to malaria, Plasmodium, to Trypanosoma cruzi, which causes Chagas disease, and Babesia, which causes babesiosis. One of the things that we worry about as blood operators and in the general community is the impact of climate change on the epidemiology of some of these diseases. On the left-hand side is a proposed graph or a picture or map of what might happen with global warming and the movement of the Ixodes tick, which can carry Babesia, into parts of Canada which currently don't have those types of tick populations year-round. So yeah, climate change is a risk that we have to think about. to the blood supply. And the impact of climate change on parasites is yet not known. And this is another paper that we recently wrote with a subgroup. But in general, our conclusions from that paper were that the blood community should be vigilant for those impacts of climate change and understand the epidemiology of the diseases as well as the vectors that carry those agents, and with those bites or with those interactions cause people who may appear to be healthy to become infected. In 2023, this became very apparent to us when we saw small areas of endemic malaria transmission in the United States. We saw them in Florida with Plasmodium vivax. We saw them in Texas on the Texas-Mexico border with vivax. We saw them in the Washington, D.C. area with Plasmodium falciparum. And recently in Arkansas, we again saw a case of locally transmitted malaria due to Plasmodium vivax. So in general, a variety of approaches are going to be used to make sure that we reduce the risk of blood supply. Donor screening questions, like some you've already generally seen, but specific ones for Chagas and malaria. Testing, in particularly for Trypanosoma cruzi, which causes Chagas disease in high-risk individuals. Pathogen reduction, which right now works quite well or partly well for Trypanosoma cruzi, but not for Plasmodium. And then surveillance followed by risk-based decision-making activities that you've already heard about. So here's two general questions that are asked on a donor questionnaire. Either you do it on site, at the clinic or at the blood center, or you can do it online. Have you ever had Chagas, babesiosis, or leishmania? And then specific questions for Chagas. Have you spent a total of 6 months or more for a continuous period in Mexico, Central, or South America? Were you born in that region? Or did your mother or maternal grandmother, were they born in that region as well? So Chagas in the natural world is transmitted by what's called a kissing bug or a triatome bug. And during feeding, an infected bug defecates on the human, and there's a bite site there. And the parasite will enter into the bite site and then cause disease. Usually, these may be asymptomatic in some individuals, but they can cause congenital infection, and they can also cause long-standing infections in individuals that require significant management. So again, an acute asymptomatic phase. In some cases, a person will develop a Shagoma. You can see that on the top. And then years later, if chronic infection does occur, people may have cardiac or gastrointestinal disease. We're talking about platelets today, and out of all the agents that we'll talk about, the three of them today, transfusion-transmitted cases for T. cruzi are primarily associated with platelet transfusion, although there has been a case of transfusion transmission with the red cell concentrate. And there's probably several reasons for that. We know that the parasite makes a transialidase, and so maybe there's an interaction there that's preferential. But those questions that we ask, as well as selective antibody testing, and then further confirmation of that at the National Parasitology Reference Center at McGill, probably play a role in reducing the risk. We also know that pathogen reduction in platelets has been found to be generally effective. And also, because we do leukoreduction of our cellular blood products, leukoreduction probably captures some of those parasites as well. Apart from Trypanosoma cruzi or Chagas disease, the other agent that we worry about in North America is babesia and transfusion-transmitted babesia. Unlike Chagas, we're really talking in this case about an agent that lives within red cells. So its impact on platelets is negligible, although we have had cases of transfusion-transmitted babesia where platelets were derived from whole blood products. probably through of contamination with small levels of red cells. There's several species of Babesia, and in nature, this is transmitted through the bite of an Ixodes tick. So again, ticks have 8 legs, and with that picture I showed before, you'd be thinking about climate change, changing patterns of tick movement, and then potentially the concern that maybe with the changing epidemiology, you might see more Babesia in your donor population. Again, the concern here is that many people may be asymptomatic, or they might have influenza symptoms. So like malaria, Babesia lives in red cells. Both those agents can present with a general malaise. And so that first question I showed you, are you feeling well, may get some of those post-symptomatic or those symptomatic individuals to not donate because they'll get screened out with that question. It can cause severe disease, even a natural infection. Recently, we published a paper in the Journal of Clinical Microbiology where we talked about transfusion-transmitted Babesia and the epidemiology, the risk mitigation strategies, and the regulation that's undertaken. In the US, it's a significant problem, and there's been over 200 cases of TTB. In some cases, the outcomes can be quite severe, and depending on the analysis of the population it's being looked at, those rates can be as high as 20% in that TTB population. Again, those are probably skewed to more severe cases, but it is concerning. And because of this, in the United States, the FDA has mandated or recommended that donations that occur in a series of about 14 states plus the District of Columbia undergo nucleic acid test, or PCR or TMA assay testing for Babesia. The other issue here is they said, well, you don't need to take the testing or do the testing if you have pathogen reduction technology. But for red cells currently, there is no commercial pathogen reduction technology that blood operators can use. The technology that might come along is probably going to be something based on maybe a non-UV light approach. So you maybe potentially a glutathione and a muscilin approach that might cross-link nucleic acid. At Canadian Blood Services, we did a surveillance project in 2018. And at the time, we did that with Hema-Quebec. We looked across the country using a nucleic acid test and worked with the American Red Cross. And we found that the positivity rate, depending on whether we use a nucleic acid test or serology, was 0.002% to 0.007%. And this was at the levels of states in the US, like Arizona, Oklahoma, where there was no recommended or mandated testing of the donor population in those regions. Dr. Sheila O'Brien, who's the head of our epidemiology group, helped us do a risk analysis. You can see on the second bullet point that nationally, without testing, you might see a positive donation that was NAT positive for Babesia once every 12.5 years. So again, the risks at that point are quite low. We then undertook what's called a risk-based decision-making process, and you've heard about this already. Really, in that case, you're coming up with generating a problem question. You're engaging participants that you think are going to be appropriate stakeholders. You come up with what you think might be appropriate strategies. You assess and evaluate them, and then you make a decision on what you think your organization is going to do. And this is a fairly well-established process that was developed by the Alliance of Blood Operators and was driven by a couple of researchers and scientists and other folks at Canadian Blood Services. The general consensus that we came to, and you can see that on the right-hand side, that the key thing to pay attention to is that we would generally be thinking about having multiple surveillance impacts, doing sporadic and regional screening, using nucleic acid and possibly serology, undertaking risk assessments, looking for signals, either in those regions or by our surveillance. So are there increases in the agent being identified in tick populations? Do we see increases in clinical cases in that area? and then reassessing, making decisions and reassessing. The last agent of interest is malaria. And again, we have several questions here that we can ask right up in the donor questionnaire. The first is a short-term travel question on the top. And then the second one is, in the last 3.5 years, have you spent more than 6 months in a continuous period outside Canada or the US? This agent in general, has 5 species that are thought to infect human, and there are a few that jump between primates and humans, but these are the 5 main species. Natural transmission is through a mosquito, an Anopheles mosquito. Transmission has occurred through blood transfusion, organ transplant, and needle sharing, as well as vertical transmission. And the distribution of the vector species, as well as the species of the parasites, varies internationally. Between 1994 and 2022, we had 4 cases of TTM in Canada, with the last occurring in 2022. And of interest to us is mostly the agent Plasmodium falciparum. This seems to be a concern in people who are semi-immune and grew up in areas where falciparum is endemic. In many cases, donors might not remember or recall if they were infected with Plasmodium falciparum or another species. Recently, we had the first identified case, that should actually be 2022, of malaria in Canada since the late transfusion transmitted case, or possible transfusion transmitted case since the late 1990s. And in this case, we basically looked at whether this was vertical or possibly transfusion transmitted. And at the end of the investigation, which we published in Transfusion, it was highly likely that that event occurred through transfusion transmission. So how do we prevent transfusion transmitted malaria in Canada and the US? In Canada and the US, we don't use serology testing approaches. And right now, there's no nucleic acid test approach that's being used by blood operators internationally, with the exception of a lab-developed test by one German blood operator. So really, we're looking at deferral questions. In Europe and different parts of the world, including Australia, those algorithms are a little more complicated and often involve testing using serologic assays. In that case, they're testing for past exposure to malaria and not for the presence of the parasite in the blood. So you can see those are more complicated. The risk here is that you defer people who are not parasitemic and may have recovered from malaria years ago and would not be considered infectious. There's a variety of risk mitigation approaches that we've taken at CBS that involve asking questions and then reducing areas where we have malaria deferrals as well. This approach has been adaptive and is one of the approaches we'll probably continue with until there are commercially available pathogen reduction technologies for red cells. So what are some of the key issues here? Canada and the US use more conservative donor screening approaches that rely on questions. Some other jurisdictions rely on serology and questions. And historically, this serologic methods lack sensitivity and for different Plasmodium species. They also detect past exposure and not current parasitemia. And the other issue here is that molecular tests for malaria right now, the limit of detection of the assays do not reach down to the lowest theoretical infectious dose for that donation. So why are we not just using diagnostic approaches in Canada at this point? So for the serology approaches, National Parasitology Reference Center in McGill has a serologic assay, but that's not widely used across the country. And so to do high throughput screening, it would not be feasible. There's also a lack of experience interpreting those serologic results in Canada. Rapid diagnostic tests that use antigen detection for malaria are less sensitive. And the PCR assays that are currently used in clinical settings, which are often used for speciation, but may be used up front to assist in diagnosis, are currently not able to reach down to that lowest theoretical infectious dose of possibly 1 to 10 parasites per donation. So given these, we know that there are a variety of issues with lab testing. However, there are some approaches that are arising in the future that we might be able to consider. But we have to take into account the balance between deferral strategies, testing, and then when we will get red cell pathogen reduction approaches. So what are the key takeaways in Canada and the US regarding transfusion transmitted malaria? So in our setting, the risk will likely be from people who are semi-immune. They may not be aware of their past infection. And travel risks to areas where there may be a recommendation for malaria prophylaxis, those risks are probably a little lower. They have minimal chance of exposure and higher chances of acute symptoms, given that many of those folks are not semi-immune. So in conclusion, a variety of processes are used to protect the blood supply. Those will include deferral questions, testing approaches, pathogen reduction, and then we'll use other approaches like surveillance and risk-based decision-making processes to adjust our approaches and come up with new strategies to improve the safety while ensuring we have appropriate blood products available in the system. And that's it for my presentation.